Chemicals called tastants are detected and transduced by chemoreceptors located in taste buds. Taste buds are found in papillae of the tongue mucosa. The taste bud is composed of about 50 modified epithelial cells, some of which are supported cells called subtentacular cells, and others which are called taste cells. The taste buds are found on 3 types of papillae of the tongue. First one is circumvallate papillae. A large number of taste buds are on the walls of the troughs that surround the circumvallate papillae, which form a V-line on the surface of the posterior tongue. Second one is fungiform papillae. Moderate numbers of taste buds are on the fungiform papillae over the flat anterior surface of the tongue. And the last one is follicate papillae. Moderate numbers of taste buds are on the follicate papillae, located in the folds along the lateral surface of the tongue. The membrane of the taste cell, like that of most other sensory receptor cells, is negatively charged on the inside with respect to the outside. Application of a taste substance to the taste hairs cause partial loss of this negative potential, that is, the taste cell becomes depolarized. This change in electrical potential in the taste cell is called the receptor potential for taste. The mechanism by which most stimulating substances react with the taste villi to initiate the receptor potential is by binding of the taste chemical to a protein receptor molecule that lies on the outer surface of the taste receptor cell, near to or protruding through a villous membrane. This, in turn, opens ion channels, which allow positively charged sodium ions or hydrogen ions to enter and depolarize the normal negativity of the cell. Then the taste chemical itself is gradually washed away from the taste villus by the saliva, which removes the stimulus. Taste impulses from the anterior 2/3 of the tongue passed first into the lingual nerve, then through the chorda tympani, into the facial nerve, and finally into the tractus solitarius, in the brain stem. Taste sensations from the circumvallate papillae on the back of the tongue and from other posterior regions of the mouth and throat are transmitted through the glossopharyngeal nerve, also into the tractus solitarius, but at a slightly more posterior level. All taste fibers synapse in the posterior brain stem in the nuclei of the tractus solitarius. These nuclei send second-order neurons to a small area of the ventral posterior medial nucleus of the thalamus. From the thalamus, third-order neurons are transmitted to the lower tip of the postcentral gyrus in the parietal cerebral cortex. This lies slightly lateral, ventral, and rostral to the area for tongue tactile signals in cerebral somatic area. From this description of the taste pathways, it is evident that they closely parallel the somatosensory pathways from the tongue. Taste reflex. From the tractus solitarius, many taste signals are transmitted within the brainstem itself directly into the superior and inferior salivatory nuclei, and these areas transmit signals to the submandibular, sublingual, and parotid glands to help control the secretion of saliva during the ingestion and digestion of food. Do not forget to like the video and subscribe to our YouTube channel.